when I first joined the GAO group at UMass Chan Medical School, I had a very naive understanding of AAV. I thought that whatever you design is sort of the expectations, but it turns out to not be true, right? There's a lot of heterogeneity that we see that is packaged into AAV. The heterogeneity can, of course, come from truncation events, so non-unit length molecules that are packaged, but a lot of these heterogeneic forms can also persist as contaminants that come from packaging components that originate from your plasmid, DNAs that are used during production, but they can also come from genomic DNA from the packaging cells, right? And so because, at the time that we had sought to understand these things, the sequencing technology landscape was still focused on short read sequencing methodologies. We felt that these methods were very much limiting. So we sought to employ long-read sequencing methods, right, to look at the heterogeneity of AAV. And because there were no prior publications on this, we had to sort of develop the platform on our own. And so what we had ended up developing was what we called AAV genome population sequencing, or AAV-GP-seq, which is based on PacBio single molecule real-time sequencing platform. And so many of you know that there are definitely limitations to the method, right? So PacBio is great at long reads, but they still rely on adaptering of a double-strand molecule, right? So there are many ways to achieve this. Initially, we had relied on the fact that AAV packages plus and minus strand genomes at equal populations, and so if you undergo thermal annealing of the sample, then you can form these double-strand molecules. If you're sequencing, self-complementary AAVs, which are inherently double-stranded, then you can adapter these on one end to form then the single-strand DNA circular template, which is compatible, of course, with the PacBio methodology. Then you can undergo strand displacement polymerase activity to do rolling circle replication, and then you can split the strands by strand specific consensus reconstruction. And the great thing about this is that you can cover the entirety of the molecule as well as the ITR sequences. And so this is a sort of a graphical abstract about what we can utilize this methodology for. Obviously, you can assess the composition of the full-length genomes, but you can also start to profile truncation events, right? So if there are an accumulation of molecules that seem to hone in on specific regions, especially those that have strong secondary structure or GC content, you can then start to define truncation hotspots, right? And then those potential things can lead us to better vector designs. The other thing that we can do is we can start to characterize our impurities, right? So actually, I'll show some of this work later on, but it turns out that a lot of the contaminants that you pick up, especially from host cell genomes, may be chimerics, right? Meaning that they've recombined with ITR-bearing sequences, and now they're ending up being packaged, right? And so a lot of this work is critical because we would like to know whether some of these contaminants may serve as sources of toxicity, right? And so, yeah, this is just sort of an example of identification of truncation hotspots, where if we know that the majority of the sequences are full length and we run standard gels and you see that there's one band, these sequencing technologies have the resolution to identify these truncation hotspots. So this is a worst case scenario. So actually, we had published on the sequencing of this construct, which actually bared a construct which carried two CRISPR-Cas9 guide RNA cassettes, right, which were oriented in a tail-to-tail configuration, which makes sense that this would form a very strong secondary structure. And so the outcome, of course, is that you get a high degree of heterogeneity represented by these reads that are aligned to the reference in this IGV display. So just to orient everyone to how to kind of read these things, because I know a lot of people don't like using IGV, but I still think that these type of displays are useful. So reads that are aligning perfectly to the reference are in gray. Those bases that aren't matching very well are labeled in the colored bases that are not matching. And then different speckles, like, you know, if it's black, then it represents gaps, and if they're purple, then they represent inserts, right? So you can get a great deal of information from these type of investigations. For this construct, namely, turned out that this construct worked, right? So you can ask, well, if it's so highly heterogeneous, why do these constructs work? So it turns out that due to the design, these sequences were actually, these vector genomes that were being packaged encompassed enough of the functional aspects of both the guide RNA cassettes, right? So that it functioned well, right? So you can still run gels and get a very high heterogeneity, but you know, if these were sort of the outcomes, and the FDA says, well, we see a lot of truncated, you know, sequences, you can use these type of technologies to then tell, you know, report to say that these are still functional, right? But obviously, you know, you would like to redesign this construct, which we did, to produce vector genomes that are more homogeneous. So in terms of understanding heterogeneity, I put together this very basic table of at least the species that we know exist based on the long-read sequencing technologies that we use. So panels A and B represent what we would consider unit-length molecules, right? So if you're designing a single-strand AAV with intact ITRs, you would pick these guys up, right? If you had a self-complementary design where you have one of the ITRs mutated, you would then be picking up these self-complementary forms. One of the major forms that we see if there is a high degree of heterogeneity that's due to some region within the genome that causes truncations, we normally pick up these truncated snapback genomes. So this term snapback genome is actually a historical definition that you know, was observed very early on, where you have standard replication, but then if it hits a region of complex sequence, then there's polymerase stalling, and then you get a switch of the polymerase back to the newly synthesized template, and then you get these sort of snapback events where you have these now truncated molecules that are self-complementary in size. We also pick up truncated single ITR sequences, but these are much less represented because of the fact that these are very hard to adapter, right? So remember that for PacBio sequencing, we require that you adapter double-strand molecules. So even though these are found in the population, they're very, very rare. But doesn't mean that they don't exist, right? The other sort of forms that we also pick up are oversized dimers or oversized partials, right? So I'll get a little bit into this because, you know, one of the things that we've been seeing more frequently during, you know, with current sequencing data sets is these giant molecules, right? So we pick up these 9 kb sequences that exceed the packaging size of AAV, which is 4.7 kb. And the argument is that, you know, these shouldn't exist, right? And it's true, they shouldn't. But why do these appear in our sequencing platforms? And so I'll go over that. We also pick up these truncated unresolved ITRs, so it has these sort of composition of, again, a double-strand molecule that's sealed on one side, but they're very different from the snapback genomes, right? And they have this unresolved ITR here, and then a double strand on one side. We also get internal deletions, and the other two populations, where I guess, two standard populations that we see very abundantly are reads that encompass sequences of the backbone plasmid, right? So these can come in two forms, either read-through genomes, where replication goes past the ITR on the plasmid and then it starts to read into the backbone, or you get reverse packaged genomes, right? And I would argue that aside from truncated forms, that these are one of the most abundant forms that you will get. And actually, because of the nature of these types of reads, you always get higher representation of reads within the backbone that are closer to the ITR. So if you ever look at sequencing summaries of alignments across the entire plasmid, you always get sort of like a die-off at the ends, right? We also see package-related fragments, as I had mentioned before, but I would argue that, that these sequences on their own are very rare, but when they are recombined with ITR sequences, right, and persisting as chimeric genomes, these are highly abundant, right? You know, we can pick up sometimes up to 2%, 3% of the entire population that would represent chimeric genomes and contain some host cell genomes as well as plasmid backbone sequences. And then there's RCAAV. So for those that, you know, run production cores, you know, replication AAV, replication competent AAVs are the bane, right, of the core, right? And I'll talk a little bit about this. And then, of course, there's also adventitious viruses. So what I wanted to do was to focus on a publication that we had submitted to Human Gene Therapy in collaboration with Magali Budlu and Edward Aoso a couple of years back. And in this work, we had basically compared the heterogeneity in AAVs packaged in two different systems, one being the triple plasmid transfection system in HEK293s versus the baculovirus SF9 platform. And the reason why this study was intriguing is because we found many interesting aspects of packaged AAV. One of the first things that we did was we wanted to know the composition of what was observed within our high molecular weight species following cesium chloride purification versus those that appear within bands that are intermediate between the full bands and the empty bands here that we've sort of called as partials. Because one of the things we wanted to do was that if AAVs are packaging indeed heterogeneous forms, what do they sort of look like, right? And at the same time we were doing this because we had the capacity to also isolate these empty capsid fractions. We wanted to sequence those as well. And amazingly, we were able to find that empty capsids aren't actually empty. They have stuff in them, right? And it turns out that a lot of the reads actually map at the ITR. So they're filled with ITRs, but they still, after gradient centrifugation, they still package AAV components, right? So that was the first intriguing thing. The second thing we found was that when you compare the full fractions from the HEK preps versus the SF9 preps, we see that the SF9 preps have a high degree of heterogeneity. And again, based off of what we saw by IGV display, we saw something that was actually very different from the double-strand truncated forms that we observed within these two populations. Because remember, we had that if there is a high degree of secondary structure, you form these snapback genomes. But what we found within the heterogeneous forms from the SF9 preps was we found these unresolved truncated genomes. And so these are very evident when you look at the composition of the alignment behavior of these reads on an IGV display, right? And so where could these be coming from, right? Based off of the work that we had published, we had realized that some of these forms are coming from what are considered oversized genomes. So in this work, we had packaged a 3.3 kb sequence, right? And so in HEK293s, the majority of the species are indeed those that are unit length. But when we looked at the size distribution of those produced in SF9, we saw these very heterogeneous forms, right? And I don't think I've shown, I'm not showing here the alkaline gel, but those gels that you run, they're all very smeary, right? And there's multiple bands that are seen. And the reason why we think that they come from these oversized genomes is when we sequence them, we get these sort of species, right, predominantly. And I think the reason why they come about is because when isolating these oversized genomes, right, because even though the design is 3.3 kb in size, it may be packaging 4.7 or 5 kb species. During the library prep steps, there may be some cleavage or damage to the single-strand DNA, resulting in the in the retaining of these double-stranded molecules, which can then be adapted. If we do start and end position analysis on all of the reads, we typically see these form in peaks, right? And so here's where you can start to define how these genomes persist when combining the composition of these double-strand unresolved genomes, and then look to see where their start and stop positions end, right, or are centered. And so you can then sort of reconstruct the composition of these genomes as such. Okay, and and then when you compare back to the alkaline gels that you run, these are the exact species that we find. So when looking at your data, right, I would argue that these type of species may not necessarily exist within the original prep, but may come from artifacts that are related to the library prep steps. So now what I wanted to do was to focus our attention on ITRs, right? So ITR biology recently, because we've started to have better resolution in our sequencing data, a lot of work is now refocused back on what ITRs are doing. And I just have this sort of zoomed in portion of this figure that we published in a review in 2019 to indicate that once single-strand AAV gets into the cell, the ITRs can then be used as sources of self-primed. Because it's a self-prime molecule, then this can initiate second-strand synthesis to form this self-complementary AAVs that are double-stranded, and then this can undergo intramolecular recombination to form these stable monomers or episomes. And then they can also further recombine into these circular concatenamers. ITRs are also known to drive integration, right, in the presence of rep. And this is sort of the natural life cycle of AAV. But we also see integration of AAVs into the host cell genome from vectors, right? And of course, vectors, they don't package any aspect of rep molecules. And so a lot of investigation has gone into the mechanisms that drive integration, right? And presumably they're because of ITR sequences, right? But I won't get into too much detail into the roles of the ITRs, but this is some area of research that we're actively investigating. So more specifics about ITR biology, because we think about these night and day, I wanted to discuss a little bit about this. So the literature about the exact structures that you read, they're a little bit inconsistent. And so some of the articles that you read, it's if the B arm is closer to the 3' end, that would be considered flip, but that's not the definition that we go by. The definition is that if the B arm is closer to the open end of the genome, it would be considered flip, and if the C arm is closer to the open end of the genome, it's considered flop. The reason that we go with this definition is that when you replicate flip, you get flip, right? You don't get flop, right? And so there is no 5' and 3' designation when you consider a plus and minus strand genomes, right? And I'll go over a little bit about this in the next slide. But what happens is that once the ITR is used as a self-prime molecule, it'll generate a double strand, and this can then further undergo another secondary structure where the TRS can then be utilized can then be cleaved by rep. And what you actually get is a very unique feature of AAV, where you get these two configurations, flip and flop. And what's unique about AAV is that they have these ratios at 1:1 that are independent of one another. And so you get this 1:1:1:1 ratio of flip, flip, flop, flop, flip, flop, from flop, flip, right? Whereas the other parvoviruses, they don't seem to have this sort of equal distribution. There seems to be some bias, right? And so, again, how do these sort of different compositions form? And I think this is a little bit important because, you know, when we consider flip and flop, it can tell you a lot about how the molecule is actually being replicated in the production lines. So let's say, for example, after resolution, we're working with, we rescue from the plasmid, and we're working with this top strand, or the plus strand of molecule, which is a flip configuration as designated by these up arrows on the 5' and 3' ends of the molecule, right? And so during replication, what you form is this reverse strand. And as I had mentioned before, when you replicate a flip configured molecule, you get flip. But in order to get the opposing configuration, what happens is that you get resolution at the TRS. The old 3' ITR then becomes a new 5' ITR of the newly synthesized molecule. So these configurations then turns to flop, and you get fill-in of the top strand, right, of the ITR, and so you have two flop orientations and two flip orientations to yield a flip plus flop molecule and a flop minus flip molecule. If you repeat this over several times, by generation 2, you will cover all of the different 8 configurations of flip and flop and plus and minus strand molecules, and then by the 4th generation, you get equal distribution of all of these configurations. And so if you were to look at ITR configuration in your preps, and you see predominantly that there's a 50-50 split between flip and flop, you know that those molecules have undergone several rounds of replication during the production. But if you have compositions where it's a little bit messy, this is where you know things have gone a little bit wrong. And so what I'm comparing here are two preps, right? One from HEK293 preps, and then one from SF9 preps. And I'm showing you here the compositions of the left ITR and the right ITR of both of these species. And when you see right away is that there's a lot of heterogeneity based on length, as well as based on classifications of ITRs that are produced, ITRs on vectors produced by SF9 preps, a lot of them being these unresolved species, right, which I had mentioned in the IGV display that I showed previously. The other thing that we had indicated was that they do fall into specific categories, right? So either you have this 22 nucleotide deletion of the B arm or 22 nucleotide deletion of the C arm. We also see some compositions where we get this 11 nucleotide deletion in the C arm. Some of you may be familiar with this, and I would argue that these came about because people were sort of sharing plasmids that had mutated ITRs, and they just sort of propagated, in our groups, right? And so and that's well and good that we can sort of identify the heterogeneity of ITRs, but what could they be, or what could they mean for production? Well, it turns out that if you stratify vectors that contain either unresolved ITRs or mutated ITRs, those that lack a B arm or C arm, or even these trident shape ITRs, they're attributed to those vectors that have unresolved genomes, right? So what this indicates is that it's very important to know the composition of your ITRs on plasmid. And actually there was a talk that was given at ESGCT this year that indicated that even if there were some, small percentage of ITR, mutant ITRs in the plasmid preps, that they can actually make an impact on vector titers, right? And so, now what we're doing is going back, in our lab at least, to resequence all of our plasmids to ensure that all of the ITRs are indeed intact before we produce vectors from them. So what also happens when you have these mutant ITRs or unresolved ITRs? And one question I get a lot of times is I see a lot of these preps with multiple ITRs, and where do they come from? And I try to do my best, sometimes we have these meetings on Zoom where I'm drawing out these molecules in a very poor way, but I put together this slide to indicate what may happen, right? So let's pretend that this was the 3' ITR that should be resolved, and what happens is this molecule then becomes this essentially longer, dimer form of single-strand AAV. This can then replicate, and you form this, right? Where you now have 1, 2, 3, 4, 5 ITRs. And if the molecule continues to be unresolved, you just get these very gigantic reads, right? Now, obviously, these don't get packaged, right? And so you can imagine that if you have mutated ITRs and you get a hit in titers, this may indicate or may be a source for why there is this relationship between mutated ITRs and poor titers or poor packaging. Okay, so if you have some of these reads where you see multiple ITRs, take a look at the compositions. If they have this sort of forward, reverse, right, whether they have some chimeric species or whatnot, these are typically the sources of it, and they're not due to, ligation events that may be happening in library prep stages, right? I'm not saying that they don't happen, but if you do this A-tailed adaptering, right, that should limit this fragment-to-fragment annealing of your libraries, right? The other thing that we've learned about mutated ITRs is that they do typically repair. So what I'm showing you here are 2 ITR configurations, one being the 11 nucleotide deletion that we published on this work a few years ago, indicating that even though you have these mutated ITRs on plasmid, they're typically repaired, right? So what happens is that it uses the wild type sequence as the template for repair in this classical panhandle configuration of the AAV genomes. And the 22 nucleotide ITRs also repair themselves as well, but this and this is work that remains unpublished. But it turns out that some of the repair mechanisms may be dependent on sequences within the construct itself, as well as the capsid that they're packaged with. And this is ongoing research in our group. One of the other things that we learned is that the abundance of recombination can actually give you these very fragmented and very complex compositions. What I'm showing you here on the bottom is a AAV vector genome that contains sequences from human chromosome 5, a partial AMP resistance gene, vector backbone, another segment from chromosome 16, and some fragments from the capsid sequence. So you can imagine that a lot of these sequences that you see where you have these very highly rearranged species, these can be potentially attributed to that. And now that we're also using long-read sequencing to look at integration events, some of the integrations that people are now publishing on also have rearrangements. So the question is, are these rearrangements happening within the integration events itself, or does it actually originate from the vector? And that's something that a lot of groups are actively pursuing to understand. The other thing about these chimeric reads is that, as I mentioned before, they are attributed to the majority of the host cell genome contaminants. So I just selected 3 independent libraries where we're showing where these reads are mapping across the human genome. We map to the human genome, of course. We use HEK293s as a packaging cell line. And as you can see, there aren't any particular hotspots, right, attributed to any different prep, right? These events are more or less stochastic. If there are hotspots, one thing that I would highly recommend is to ensure that the vector that you are working with doesn't contain aspects of the human genome. And if it is the case, then these regions have to be masked, right? And so now I wanted to start to discuss sort of the other types of contaminants you may see, one being RCAAV, right? Which I think the FDA requires that labs assess the abundance of replication competent vectors. And a lot of groups that I've worked with, they just ask a simple question, right? Can we detect full-length cap in the preps? And I would argue that may be a limited way of detecting RCAAV. Number one, even though the read depth of PacBio is really good now, it's not like you can see every single molecule within the population. The standard method for assessing RCAAV is to take the prep and then passage them in the presence of adenovirus, upwards of 3 times in cells, and then assess by qPCR. What we did in this study that was published a few years ago was to use PacBio to determine how these recombination events are occurring. And it didn't seem to have any specific sequences that define these recombination events. So these recombination events may happen randomly, and if they do start to overpopulate your preps, right? After this sort of configuration, these are the sources. But I would say, the argument to the detection of these is, are these actually being produced while we pass these through different cells, or did they originate from the original prep, right? And that's again a hard question to ask, because we do see rep and cap sequences in PacBio, right? A lot of the reads, majority of them, I would argue, are not full intact rep or full intact cap. I think, doing sequencing projects with many of you and with other collaborators, even though we do pick up rep and cap, I've never seen full rep and cap in the original preps, right? And so I wanted to end with this slide because, I like the idea of the definition of a chimeric, right? In Greek mythology, it's a fire-breathing monster with a head of a lion, a goat's body, and a serpent's tail. But the other definition is illusion or fabrication of the mind. And so when we first started describing these, chimerics, people say, "Oh, these are artifacts, right? You're getting ligation artifacts, and they're not real." But I think we should start rethinking these sort of things. We don't want to bury our heads in the sand. And if there is going to be a push towards understanding toxicity and integration events that carry a lot of these recombined genomes, then understanding how chimerics form is going to be important for us, right? And so with that, I'll have my acknowledgement slide. So some of the work that I presented here, again, was in collaboration with Magli Budlu and Edward Aoso at INSERM Institute. And a lot of the current sequencing work that we do is in collaboration with the Deep Sequencing Core that we have at UMass Chan Medical School. And then we also wanted to thank PacBio for their constant support throughout the years. Thank you.
I think we have time for one question, if there's any questions in the audience. Otherwise, yes.
Okay, so on the flip and flop, like the ITR characterization, how do we use that information? Are we using that information to design better ITR sequences? Is that what
So currently, the ITRs that are being used in all production platforms are based on AAV2. There have been several groups that have been designing synthetic ITRs to see whether they perform better. Arun gave a talk at ESGCT describing his Gen X, Gen Y, and Gen Z ITR sequences. But one of the things I think those designs are limited in is production, right? So they may have enhancements in transduction when you deliver them into mice and in tissues, and also in cell culture, but they tend not to package well. And it could be because even though they do perform better in transduction, maybe they don't replicate as well, right? And so I think part of using PacBio and these long read sequencing technologies may be to start to understand that. And in saying that, we are also very much interested in exploring other ITRs. And so I gave a talk 2 weeks ago at ESGCT describing new ITRs that we've identified for AAV8 and RH39.